Good day, everyone. I'm Ivan, the moderator for this webinar. I welcome everyone to today's webinar, which is Life Cycle and Validation of Analytical Procedures, FDA, ICH, and USP. I want to introduce our presenter for today, John C. Fetzer. John C. Fetzer, PhD, brings over 30 years of experience in good laboratory practices, working as both a method developer and lab supervisor. He has led numerous training courses and webinars and has also served on the editorial boards of Journal of Chromatography. I would now like to hand over the floor to the speaker. Over to you, John.
Good day. Today's webinar is on life cycle management, which has been part of good laboratory practices informally for a long time. Why is life cycle assessment useful and important? There is much jargon, as with many things GLP, with life cycle management. It is those criteria that define the validation of a methodology. Why are they doing this? Because the concentrations that are being used are a million times more. How to do a life cycle assessment? You look at the methodology and you look at its purpose. Main principles of life cycle management are spelled out as a combination of these two ICH documents that are readily available, ICH-Q2R2 and ICH-Q14, describe the recommended development and validation during the lifetime of an analytical methodology. The main principles of ICQ14 are giving descriptions to method development and risk assessment. This is just a concept of the analytical procedure lifetime. Basically, the keys around if you have a change, if nothing changes, there's no problem, method is still good. But when you have a change, you have to assess what that change has or will have on the procedure. This ICR Q2R2 is the one that gives guidance on how to develop a method and decide on a methodology. You have to have a validation study, which is basically looking at procedure form characteristics. These are some possible criteria. Once again, they are just possible criteria. They are not a checklist. You choose the ones that are applicable to the methodology you're using. Specificity is a very broad category, but it basically means the information that you have is definitive enough so there are no interferences, no matrix effects. These are just some more details. When you're doing identification, you have to make sure that there are no interferences. Linearity is one that many laboratories fail on because basically a large proportion of laboratories only use correlation coefficient. Accuracy. Accuracy means comparison to something that is acceptable as having a known value for quantitation. Validation range is the range that the data is going to be reported. Validating precision can be very, very, very difficult. It depends on what the purpose may be. There is the overall precision of the measurement, which is just the plus and minus in the value if you're doing a quantitative analysis. Repeatability is just the precision, the amount of plus and minus, if you're trying to do everything exactly the same. Limited detection. Limited detection is problematic because formally, all the chemical societies, including the International Union of Pure and Applied Chemistry, define limit of detection in terms of signal to noise of a methodology. A limit of quantitation by definition is similarly, it's 10 times the standard deviation of the methodology of a blank or 10 times the baseline noise. Stability is one that many laboratories do not comply because they focus on stability of the samples. Once again, they are not a mandatory listing. You have to choose the ones that define the methodology and show that it works. ICR or ICH has no definition of ruggedness. You use all these things to gauge a methodology's lifetime. Archival samples are something that's recommended in particularly things like purity analysis. Trend monitoring is part of all that. You avoid reevaluation with each change in a methodology that could have been predicted. These are just some things to be aware of. And with that, I will close. I'd like to thank everybody for their attention.
Thank you, John. That concludes the session for today. And thank you everyone for participating. You may all disconnect now and may you have a great day ahead.